I could talk about something else also, but I thought let us make it connectomes only. And why? Because if you want to understand connectomes, you have to, you can use network theory, and you have already studied, have been introduced to the network theory. So how to use network theory to understand the connectomes, that's mostly the part of my talk. And so to start with, so what is connectome? So basically the connectome is the map of the nerve cells. And so when you're thinking about the brain, what brain has? The brain has this, the nerve cells, and they are connected through the gap junctions or the synapses. And so in the way they form a network. So like the cells, they process the signal, then they send that information to the other cells through their connections, which are mediated through the synapses or the gap junction, which are the direct connection between the cell body. Okay, so in a way they are forming a network, because there are two main components of a network, nodes and edges. So you have both here, nodes and edges. Okay. However, the way I described the brain network was considering the complete cell level connectome, which is about the connection between the neurons. And for that, the complete cell level connectome is only available for, I think, the C. elegans neural network, which has around 316 neurons approximately, and the fruit fly brain, which is having 1,40,000 neurons in the neurons. So far the full connectome that we have is for only these two. For other, we have for different brain regions, we do have the cell level mapping, but for these two species we have full complete cell level connectome. So if we do not have the cell level connectome, what we have for the other species? So what we have are either the structural connectome, where different nodes are not the cells but the different brain regions, and the connections between them could be the nerve fibers that are connecting those brain regions, and therefore called as the structural connectome. So this structural connectome is basically the anatomic, is coming from the anatomy of the brain. So the methods to construct structural connectome is either the brain parcellation or the other method is DTI, which is diffusion tensor imaging. And the another way to construct the connectome is through connectome, the functional connectome. Again, nodes here are the brain regions, but the connections between them are the correlations. So these are studied through the time series data. So the, like in EEG and MEG, we get this, the time series of the fluctuations of the different brain regions, like through the electrodes, and they study the correlation between these time series, construct a network on the basis of it. And because this the correlation would be there because there is some functional connectivity between them, the function is getting affected by that brain region, or they are, if they are completely correlated, meaning that they are doing same functional work, okay, at the same time. So therefore these are called as the functional connectome. This is mapping the function and this is mapping the structure. Okay, so we have the two methods by which for the higher species you can study the brain networks by constructing the connectomes. Okay, one more way is there, like I think is through the gene expression data. Like now one more approach is there. I don't have it here, but I wanted to introduce that too, like having the gene expression data from the different brain cells and considering the constructing the network out of that. So those genes that are expressing in this like same cell at the same level means they are connected somehow. So once you have network, you have to study the network properties, and you have already been introduced to the network properties, like if you have node, the number of the neighbors or the degree, and then the clustering, and then modularity. So here, like how all these properties are associated with the understanding the brain. So first is like these network metrics, clustering, motif, modularity. This may help you to understand the segregation in the brain. Okay, because the clustering talks about how the neighbors of neighbor are connected. Okay, so it gives you an information that whether there is segregation or not. Higher clustering means there is segregation. Okay, again motif, the same thing, and modularity is also the same. Thus the difference is that the higher clustering you will see in a sparse network only if it is a modular network. And the network properties such as distance, path length, efficiency. So you must have been introduced to distance, path length, the shortest path between the nodes, and the inverse of the path length, which is the efficiency. These all tells you about the integration in the brain, like how different brain regions are integrated and therefore working efficiently for the processing data. Okay. And then influence, because if you are talking about the brain, in the brain the different regions could have a different level of importance. So which brain region is more important to it means which is more influential. To study that, you can study either the degree is how many neighbors you have. So higher the number of neighbors you have, more influential you will be. So a brain region which is more connected to the different brain regions would be more influential because it is giving information to many other brain regions. Okay, and the participation coefficient is again it is about the modular structure in the network. So the nodes which are having higher participation coefficient, they have higher participation coefficient value in a modular organization. Okay, so they are giving more connection to the different modules than the assigned module. And betweenness, you already know, betweenness is the number of times a node is coming in the shortest path between different pair of nodes. So higher the betweenness centrality, more important would be that node. So in that way you can understand what are the brain areas that are more influential. Okay. Okay, next is topology. So if you want to understand that so once you have the brain network, you want to understand how is the topology of the brain network. So these are the network metrics that helps you to understand the topology of the brain, like degree distribution. So you already know that through the model networks, like the random networks, Aradhana random networks show you the Poisson distribution, and the scale-free networks have the power law distribution. So by just looking into the degree distribution, you can tell about the topology of the brain. Again, the communities may help you to tell that whether there is segregation or not. And the rich club is basically the group of the nodes that are dense, that are high degree nodes as well as very densely connected between themselves. Like in this schematic you can see these red nodes are forming the rich club. They are high degree nodes and are well connected and therefore forming the rich club. And the core, so the core nodes are again mostly the high degree nodes, but they are connected in a way that they form the core of the network. It means that they are mostly connecting to the most of the nodes in the network, either through their neighbors or somehow, but they are forming the core. Okay, so like in this node is not the core and the rest of the nodes are forming a core. And another is the fractal dimension index. So like as you know, the fractals exist in the nature. I will discuss it again. And a brain topology can also have a fractal structure. So whether there is fractal structure or not, you can find it out using the fractal dimension index. And fifth is robustness. So how robust a given brain network is, to study that you can study the percolation. You know that how a given information or any error spreads into the network. In terms of physics, you can think of like if you drop the ink on the floor or surface, then how it's percolate through it. Okay, so it's so similarly, like if there is a problem in the brain, some error is there, then how that error is propagating in the brain, that you can study using percolation. Again, degree distribution may help you to understand the robustness, because you see the power, in case of the power law behavior, you have very low high degree nodes, which shows that the network will be highly robust, you know, or with respect to the removal of the random removal of the nodes, whereas a targeted removal will highly impact it. Okay, so if you just see that if a network is following the power law, it means it is going to be fragile with respect to the targeted attack, because it has more less high degree nodes, but it is going to be at the same time going to be robust, because with respect to the errors. Why? Because the second moment of this power law distribution, this diverges. Okay, so somehow your network is not bounded, you don't have and you have scale free structure. Okay, so therefore there is a robustness with respect to the errors, and studying degree distribution can give you that idea. Okay, so you have already gone through this thing that the brain networks are modular. Yes.
When you say fractal dimensions,
Yes.
is it some kind of an embedding, like you're trying to embed the network in some
Right.
dimension?
That is through the time series analysis. Right.
Exactly.
It is about that how many dimension the network is embedded, like. So it's through in like, so once you have the time series, what's your purpose? Your purpose is mostly to come up with a model that fits to the time series. So what would be the dimension of that model? It means from what dimension that time series you have generated. For that you calculate. So if you have the time series data from the brain, you can do it for it as well. Okay. Okay, it went. So I was saying that you have already gone through it, that the brain networks are modular, and so like the C. elegans brain, it has also been shown to be the modular. Almost all the brain networks that we have studied so far are modular, whether C. elegans or rat or human, or whether it is the structural network or it is the functional network, all the brain networks has found to be the modular. Okay. And so why to be modular? A modular organization provides the independent specialized subnetworks. So like this is one of the ideas which was formulated by Simon, that nearly decomposable systems that are built of sparsely interconnected modules, for them it is the faster adaptation is possible. Okay, in response to the changing environment condition. It means the system may adapt with respect to the environment without changing the entire network. Okay, because because of the modular organization. Okay, so in a way you can understand the modules are what, the group of nodes that are densely connected within themselves and sparsely connected with themselves. So if they are densely connected within themselves only and sparsely connected with the other groups, and if there is any changes that is happening in this group, then it won't immediately affect the another group. Okay, and this is one more thing. This is again in adding to the previous statement, that the modular organization provides the time scale separation, like the time scale at which the modules are working and the entire system is working is different. Okay, and again robustness, like if in a module there is a error, it may get localized to that module and might not propagate to the entire system. Okay, and again the modular organization provides the coexistence of segregation and integration, like the segregation is required We will see how again. And integration is definitely required. Why? Because integration is associated with efficiency. Time scale separation. Okay, this is okay. This time scale separation has been proved in terms of this the propagation of diseases. So this is from the Citabro's paper only. They have also worked for the different set of the network where we had 3 time scales instead of the 2 time scales. Yes, so if a disease is spreading in a network, it may remain localized to a given module, or it will take some time to reach to the another module or community, and by then you will get some time to, you know, face that or how to deal with that disease. Okay, like the vaccination or the precautions that you have to take. Okay. So about the efficiency, how the efficiency do. So just studying the efficiency of the connectome can give you information about the brain. Is that how smart you are? Okay. So like a higher efficiency has shown to be associated with higher IQ, and it has been proved for both the structural connectome as well as the functional connectome. For the structural connectome, which was obtained from the GTI, and they found that for the binary network and for the weighted network both, this the global efficiency was correlated with the IQ, different IQs measured. Okay. Similarly, here in case of the functional connectome, it was found that this is for the path length. So negatively correlated with path length, and path length is inverse of efficiency is inverse of path length. So it means it's positively correlated with the efficiency. So again, the IQ here in the functional network is shown to be positively correlated with the efficiency. Okay. Whereas at the same time, clustering doesn't show this feature. Okay. Why? Because you know that the clustering feature is there for the segregation. For the integration, it's efficiency which is important. Okay. Yes. Path length is the shortest path length between. So efficiency is summation of inverse of shortest path length between all the pair of nodes in the network. Shorter are the path lengths in the network, higher would be the efficiency. Path length means the time and information, the minimum time and information will require to reach from one node to the another node. Okay. So shorter is the path length, faster would be the propagation, more efficient would be the network. Okay. So this has been shown for both the structural as well as the functional connectome. Human. Yeah, because IQ, you know.
What exactly you mean by IQ? Like what exactly?
Okay, so this, see, you have given the IQ test, etc. So there are set of the equations or the set of the questions that has been asked, and for that given answer that the IQ is computed. So
Okay,
you have given the IQ test, I think. You know, online it is available.
IQ is like testing a very
So suppose, so what they did, they checked the IQ. So that's why they did both. So this is one of the thing that you can study, and the network science has something to offer there to give some correlation. Okay.
Is this the brain?
That could be a debate topic that, but they show a positive correlation. So yes, and this is shown for both the structural as well as the functional. So yes.
Is the brain areas?
This is for human. For mammalians, we don't have the
Brain areas connectivity or
brain areas. For mammalians, we don't have the single cell, complete single. Okay, so this is the, if you are more interested, you can go through this reference. Okay. Okay. Now one more insight that the network theory has about the brain, that the brain has the economical trade-off between the wiring cost and the efficiency, because brain is a spatial network, so it comes with the cost. Okay. So the manufacturing cost, which is the, you know, associated with the cells, as well as functioning cost, like the each cells required some energy to work. Okay. So, so there is a trade-off between the wiring cost. So the purpose is to minimize the cost and maximize the efficiency in the given or in the least cost get the most efficiency. So it was done by one of the groups to see whether we can rewire the connections to get the minimum cost at the same time getting the maximum efficiency. Is it possible? So the answer was no. If you try to minimize the writing or the length of the connections, it maximizes the path length. It means reducing the efficiency. Okay. So for both the macaques, C. elegans, it was shown that the brain networks are optimized for the lowest cost and the highest efficiency, and there is no other way to come up with a network which is having higher efficiency at lower cost than the given network has. Okay.
In some sense, they have experimentally minimized the.
No, not experimentally. Then it is like you have a network, rewire it for the for the optimize. So it's a optimization problem. First could be the optimize the cost and see what is the efficiency. Second could be optimize the efficiency, see what is the cost. So anytime you don't see that there is a better network than these real world networks. Okay.
Just a descriptive question, but how is it rewire? Like,
how you rewire the connections?
yeah.
Randomly. So there could be many strategies actually when it is about rewiring. One is the random rewiring. Okay,
So these are from simulations. Right?
These are from simulations. So rewiring is from simulations. You cannot rewire the. Okay. Rewire is from simulations. Right? Okay. Now one more work that shows that the higher centrality means providing better efficiency, which is coming with the higher cost. So the brain, so these numbers are the brain regions, and the y-axis is showing the glycolytic index for those brain regions. It has been found that it is correlated with their centrality rank. So higher the centrality of a given brain region, more energy it requires for its functioning. Okay. So once we already know that how the brain networks are, they are modular, they are optimized for the cost and the efficiency, then what model fits best into the brain networks? So you already know about the small world networks that are intermediate between the regular networks and the random networks. The regular networks have very higher values of the clustering because the neighbors are well connected, but at the same time they have very low efficiency. On the other hand, random networks are highly efficient because they're well connected to the, but because these networks don't promise that the neighbors of neighbors would be connected, therefore their clustering is low, and the small world network are in between, which gives you higher clustering as well as higher efficiency. And how to get that? To rewire the connections. So this is the model which is given by the Watts-Rogers. Sakti has already discussed this, that if you rewire these connections randomly from the regular network, then you will get for some value of randomness a network which is having both a higher clustering as well as higher efficiency. Okay. So what about the brain? Because this is about just the topology. This is not considering this thing that the networks are embedded in the space. They are associated with the cost. Okay. So with the cost also it's the same thing. You see, in case of the lattice topology, the clustering is higher, but these are really very costly. Less costly because only neighbors are connected. Okay. So the wires, if you think of an electric network, the wires that you need to connect the different circuits are smaller, and therefore the cost is lesser. Okay. And if you have a random topology, because there are connections from anywhere to anywhere, and it is uniform, there is no biasness. So there would be as many long connections as short. So all the connections will have uniform distributions with respect to their length. So these networks will be highly costly. So though these networks will be highly efficient, their cost is also very high. And for these networks, they are less costly, but again they are not fulfilling the purpose because the efficiency is low. Okay. So the small world networks again comes here, where now on the basis of length of connections you have a small world architecture. Okay. So like yes, you want to say something? I think you want to say something. Yeah. Okay. No. So we are here to listen you if you want to say.
I know.
Then don't interrupt me. Okay. Okay. So here you have again the both features. This high clustering as guaranteed by the lattice topology and higher efficiency as given by the random networks, but they do not have all possible long range connection. They have some long range connection which provide the fastest communication between the distance brane regions, and therefore at lesser cost, higher efficiency. Okay. So if you want to model them, you have to model them with the networks, the length of the connections, the probability of the length of the connection should be proportional to the okay. So that you have to think now. Okay. Okay. So this was up to about the model. So how to model the brain network once you have the network and you want to understand it more with respect to the model network, then what model to choose? Choose a special small world network. And now I would talk something about the fractal topology in the brain network, which is also something interesting to do. If you have a data, what to do with it, you can also study the fractal topology. So like what are the fractals in nature? I think you must have studied fractals. So some of any one of you had any course where fractal structure was taught? Okay. So okay, so I will then tell you from the very beginning the fractal structures. Like the name fractals were coined by the Mandelbrot, who was first studying the length of the coastal of the Great Britain. So coastline, the length of the coastline. So suppose you have to find out the length of this or the width of this classroom, how would you calculate? We'll take a ruler, and then with that ruler you will see that what's the length. Now suppose you take another ruler which is of twice length as the ruler that you took last time. Again you will check the length, and the length will remain same, isn't it? Should remain same. It's irrespective of what ruler you take, it should remain same. But what Mandelbrot got that this for him this the length of the coastlines was changing when he was considering a step size of different length. Okay. So here you see this is the step size epsilon. So suppose he took some epsilons. So this is a logarithmic scale, and then he considered the length of the coastal line, and then he once he reduced it, the length of the coastline increased. Okay. And there was a correlation between this, there was a scaling between this step size and the length. Okay. So what does it shows? That maybe the way he was calculating the length was incorrect. Okay. Maybe the coastline does not have the unit of length. So this was his thought. So what it has then? So later on he came to know that, or he gave this idea of the fractals, that the coastlines are not having the unit of length are not one dimensional system, they are fractional dimensional system, and their dimension is around in between 1 and 2 based on the different coastal line you have. Okay, and from then the lot of study has been done on this fractal topology, and in real world you always see that like the example of this leaf, or this is the example of tangled nanoparticles, this is the example of our lungs, which are all having the fractal structures. Okay, so what are the properties of the fractals? That they have self similarity, they have irregularity, they have infinite detail, and they have fractional dimension. Okay. Okay, so in nature we get a lot of fractional, so the fractal structures. In mathematics also many, the fractal structures, the way to create a fractal structure has been proposed. So one of them is this Koch curve, and to find out the fractal dimension, one of the method is the box counting method, like cover the given structure with the box, then see how many. With the one box, of course, it would be covering the entire structure. Then reduce the box size by half. Then see how many boxes are covering the given pattern, and then keep reducing it, and then study this log of n, which is the number of the boxes versus number of the boxes that are covering that pattern, and the size of the box. If you get such a correlation, it means the dimension is fractional, and the slope of this curve will give you the dimension of that structure. Okay, so like here for the Koch curve, the dimension is 1.28. Okay, so in the case of brain also, it was found that it has the fractal topology, and in brain, how do you do so? To study the topology or find out the dimension of the topology of the brain, you divide the brain network into the different boxes which are having the different nodes, and count the number of connections it is having. And if you plot the size of the box with the number of the edges it is having, you see that it's a straight line. It will give you an idea about the fractal dimension or the fractal structure of the brain topology. And this idea came from, again, one more IBM employee. So Mandelbrot was also IBM employee. Okay, so he had given this the Rent's law. His name was Rent's. He has given this Rent's law that the number of pins that you have from the IC chip is proportional to the number of scaled with the number of components that are there in this IC chip. Okay. So following the same idea, this was studied, and it was found that in brain also. So here this is for a VLSI design. You see it is having it is correlated with the number of the box, the size of the box, and the size of the connections, or the number of the pins it is having. Same you get for the C. elegans network, human MRI, and human DSA.
So just a general sort of just to clarify, so in that figure which you had in the previous slide where you had like the different boxes around the next one. Yeah. So here since you're looking at the number of connections crossing the boundary, then I suppose that it will not matter what the layout is. You can have like where you place the box.
Right. It should just measure the size of the box, number of the nodes in the box.
What is P stands for?
That's the probability coefficient. See, sorry. Number of pins. So number of in IC. Have you ever seen an IC chip? No. Okay, so I cannot explain that. So if you see an IC chip, you'll see there are different pins. Different pins means those many connections that IC chip can have. Okay.
How would you define terminals for brain networks?
Okay, so for terminals of the brain networks are the number of the edges that are going outside that defined box. Okay, that's how it's coded. Is here these are the numbers. In case of circuit, these are the number of wires. Number of, here again wires, but again the biological wires. Okay.
G is the number of boxes. Components.
So in this chip, how many components are there? The IC chip will have what? So I am just explaining in this this thing more than the brain. Okay. What has IC chip? IC chip have many diodes, transistors, etc. So number of components, number of such units that are there in that IC chip. Okay. Okay, so once we know that, okay, given a brain network, what we can do, we can also study the fractal topology. Next, given a brain network, you can also study, you can also have some insights from the core analysis. So as I mentioned that earlier, the core may help you to understand, to have an idea of the topology, as well as it gives you some idea about the robustness. Okay, because generally the core acts as a hub, because the core nodes are the nodes that are very well connected within themselves as well as all the nodes in the network. Okay, somehow either through their next neighbors or so then that's how they are forming the core. And so like so do you all know what is the core structure? Some of you know. Okay. Okay, so here you can see with this schematic that these 1 degree nodes are, these are the peripheral nodes. Okay, so you remove these nodes, you are left with these nodes, lowest degree nodes. You again remove these nodes. In this process, if you keep doing, you get the core of the network. Okay, and so it has been found that the from the study of the C. elegans network, it is found that the core comprises the sensory and the motor neurons. So maybe the sensory and the motor neurons are the most important neurons for the C. elegans, and therefore they are forming the core, so that if there is any problem in the other system, it doesn't get affected, or somehow it is integrating most of the brain areas. Okay. And in the human cortex, the core regions are associated with the consciousness, and also it has been studied like that in cognition the core is important, and therefore there is an impact of age on the core. Okay, so as we age, how the core changes, all these studies has been done. Okay, so this is how one of the things. Soon we will come to our work, like this is our work with the Pratik. Pratik is here. If you want to have detail, you can discuss with him. So we have also studied the core organization in the different developmental stages of the fruit fly. So as we know that for fruit fly we have the complete connectome. So for fruit fly we have the complete cell level connectome for both the larval and the adult brain. Okay. So here we got an opportunity to study the difference in the topology of these two stages of Drosophila. So like in the larval brain there were 2952 neurons. In adult brain these are the number of neurons in this strongly connected component. Again I'll have to tell you what is the strongly connected component. It will take time. Okay. The strongly connected component is the part of the directed network where you have a path between each pair of nodes. Okay. And so we find that in case of the larval brain, the mushroom body neurons form the core. So what are the mushroom body neurons? Mushroom body neurons are the the part of the brain of Drosophila which performs most of this computational thing, processing the information from the antennal lobe and other brain areas. And so So here we got that whatever, so all the core neurons were the mushroom body neurons, and all these neurons are also, these core neurons were also forming the rich club. As I discussed earlier, the rich club also gives us information about the integration in the brain and the topology of the brain. Why rich club is associated with integration? The rich club are what? The nodes that are the high degree nodes, and they themselves are well connected. Okay, so they work as a hubs that are communicating faster information between themselves. You can think of it as like the transport network like Delhi, Kolkata, and all the metro cities. If they are well connected, it means there would be faster transportation, isn't it? So same way you can think of the rich club. Rich club are the brain areas that are high degree nodes. It means they are important and they are well connected with themselves. It means they are connected for the faster propagation of information throughout the brain. Okay? And so we see that the core neurons are also forming the rich club. And in case of the adult brain, we see that now the mushroom body neurons are not forming the core. Instead, the antennal lobe neurons are forming the core. The antennal lobe neurons are basically associated with the olfaction in the fly brain, fruit fly brain. So now you can see there is a shift from the core structure in the this fruit fly brain, and from just studying the core we can say that okay, there is a change in the network topology. Okay, if you see the other properties, network properties might be same, like both are modular, though this network is more modular than this one, but both are modular, but there are differences if you see at the core level. Again, the rich club organization for the core networks. So here we see that the core is not forming the rich club, and this was the question we wanted to answer, Pratik, that why here the rich club neurons are not. So one of the the paper, the data which the data was published by some other authors, and they had published that these high degree neurons are basically the these neurotransmitters that are giving this negative connections. What are these called as, Pratik? GABA neurotransmitters. Yeah. So that's why they have less connections between themselves, and therefore they are not forming the core. This is one of the explanation that was given in that paper itself, but we had also found that the rich lobe is not present in the adult brain core. And next, as I had discussed, we can study the degree distribution to tell what is the topology. So we had also studied the degree distribution, and we see that they could be same, but here it's not same. Okay. So here we see in case of the larval, it's following the Weibull distribution. So it's having the features of both the power law as well as exponential distribution. And in case of the adult fruit fly, we see that the degree distribution is adhering to the strictly to the power law. That too, for the in degree, this exponent, scale free exponent is 2.96. If you know about the scale free networks, so a network may follow the power law distribution but may not be scale free. Scale free networks do have the exponent value between 2 and 3, because for the exponent value between 2 and 3 only the second moment in power law diverges. Okay. So with respect to the in degree, it is forming the scale free network, and for the out degree, it is forming the scale free network, and with respect to the in degree, it's close to the scale free design but not in the scale free design. So here we see that clearly the topology is changing, and with using the network theory we are able to show it. Okay. Okay. Next is about the allomeric scaling in the brain. So like we studied earlier too, in case of the brain, this the body weight is correlated with the brain of the weight of the brain. Brain volume is correlated with the average number of the synapses passing through the per neuron, and the gray matter volume is correlated with the white matter. So what is the gray matter, white matter? Gray matter are actually the brain regions, and white matter are the these fibers connecting these gray matter, okay, or brain regions. And this is the the brain volume, and brain volume is correlated with the glucose metabolism. So bigger the brain, more energy it will require, okay. So we already know that in case of the brain there is allomeric scaling. So we wanted to ask this question with the help of our collaborator in this Tel Aviv University, who had the data for the 226 mammalian brains. And we were asking this question that does size affect the topology, because we had the data for the brain sizes ranging from 0.08 milliliter to 1597 milliliter. So from the brain of a bat to the brain of a dolphin. Okay, so to check that, first we check the small world property. So we studied the distance distribution. We already know in case of a small world network there would be higher local connections, short connections, and there would be some long range connections to provide the faster communication, okay. So this is the fiber length distribution, and you see that here we see for all the brains there is higher probability of the shorter fibers than the lower fiber, longer fibers, okay. So they have small world property, and also you can see that, okay, so if you see the power law distribution with respect to the fiber law with the length, it should be like this. So I should have had this, and here, so there is finite probability of the longer fibers, but not as much as is shown by these networks, okay. They are showing the bimodal distribution. They are having the longer connections way more than the expected from the model. Okay, so we were asking this question also, why we have those many of connections. I will answer to that question also, but before that I wanted to say that we also saw the rich club organization in all the mammalians that we had studied, 226, okay. And also this property that we are seeing, that the small world property, we saw that small world property is scale invariant. So that plot was for the whole brain. If you divide the whole brain into two hemispheres, like here you see it is for the 4 hemispheres, but for the 2 also it follows the same, okay. So higher probability, so this bimodal distribution. And even if we, this is for the 8 partitions, okay. For the 8 partitions also we see that this remains same, the trend remains same, that too with the different kind of clustering techniques. So not go into details of that. So if you have a brain, you can cluster it or divide into the different units with the different techniques, okay. Okay, so to answering to the question that we get that the small world phenomena, but we got that the long fibers were way more than the expected from the model, okay. So we got that the brain, all the mammalian brain comprises the 2 subnetworks. One subnetwork has the property of like complete small world, like this thing, okay, and the another part has this non-localized connection, uniform distribution of the fibers, it means randomly connected, okay. So now you see that the brain is having this property, the small worldness, but at the same time some of the brain regions are randomly connected against the rule of this, that they should strictly follow this small world property, smaller but not as much as shown by this. So this is explained by this, that in mammalian brain there is the coexistence of this small world as well as the random feature. Some of the brain, so one subgroup is following the random architecture, they are connected randomly, and another is following the spatial small world property. And so to answer that, we had gone through this, so actually we have not yet confirmed, we have not yet postulated because it is still in the process, because in case of the brain, like some of the brain regions has to have connected. Okay, so like the visual cortex and the frontal, they have to be connected irrespective of that they are far. Okay, so this structure we get from there, those brain regions, and this structure we get in general. Okay, so in summary we can say that what we learned, that the brain networks are economical, small world, have optimized for cost and efficiency. The modular organization provides that thing, that there is both segregation and integration. Also it provides a level of independence to the different brain areas. Brain has a fractal topology, and in the developmental phase of a species, there could have different topology as per the functional requirement. And in mammalian brains, irrespective of the different sizes, they have the small world topology with over-representation of the longer fibers, which are again associated with the two subnetworks in the network. And again, the small world feature is scale invariant. Irrespective at what scale you see, there is the small world feature there. It's not like that in brain there are communities and they are connected through the long spatial community, they are connected through the long connections, therefore they are forming. If it was that, then if you divide the brain into two parts, you will lose that property, isn't it? Okay. In the end, I would like to acknowledge my collaborators for this works that I discussed, and the funding agency, and to my group members, the one who is here and those who are not here. And thanks and questions.